Okay, good afternoon, everybody. Welcome to today's talk. I would like to take this opportunity to welcome everybody who's joined today for the Zoom meeting, plus people who are watching on our Facebook page. It's also being live telecasted there. Also, this meeting is being recorded, so we can use this for training purposes at a later time. Let me tell you that this is the first of its kind talks that we have planned for the Big Butterfly Month. And Big Butterfly Month is a partnership between more than 50 organizations working in the field of biodiversity conservation. And the aim of this festival or this celebration is to have India's biggest biodiversity festival, focusing on outreach, education, and interesting programming based around butterflies, which will all culminate into butterfly counts as well as sharing data over online portals like iNaturalist, Butterflies of India, and Indian Biodiversity Portal. Like I told you, it's the biggest festival yet to be organized in India, and it's happening for the first time. We couldn't have asked for a better person to open these talks. Uh, Krushnamegh Kunte, Dr. Krushnamegh Kunte has been counting butterflies since he was in high school or in college for a very long time. And he's been counting these butterflies for fun. Now he's one of India's most eminent conservation scientists, and he is affiliated with the National Center for Biological Sciences in Bangalore. He's also the man behind Butterflies of India website and the I Found Butterflies website. He today is going to train us, is going to tell us about the secrets of counting butterflies, the fun of it, and how we can use all of this data that we are going to accumulate over the whole month, and what could be the strategies going forward using this network, leveraging it to reach to maximum number of people. Before I hand it over to Dr. Krushnamegh Kunte, I would like to set out some guidelines. Firstly, I would ask everybody present to switch off their videos plus mute their microphones so we could have bandwidth and everybody could enjoy the talks. Secondly, if you have any questions during the talk, please do use the chat box to put in your answers, put in your questions, and they will be taken at a later time. Same goes for our Facebook page. People who are watching there, they can put in their questions in the comment section and they will be taken when the Q&A part of the talk will be there. So without further ado, let me bring on Dr. Krushnamegh Kunte and have him begin the talk. Krushnamegh?
Sure. Just a minute, I'll share the screen.
Please.
Okay. Can you see the screen now?
Yes.
Yes.
Great. Good afternoon, everyone. I'm really excited to speak to you today about counting butterflies. This was something that came very naturally to me. I was in 11th or 12th grade, I think, when I first started counting butterflies, and nobody actually told me to do this. I was just watching butterflies and I realized that every few months, the butterflies I was seeing were different around Pune, where I grew up. So I decided to actually start counting butterflies to document how these butterflies almost appear as a relay one after another over seasons. So this was something that really came naturally to me, and that essentially launched my career into research. And later I became a professional biologist as well. But I continue to do this not necessarily because there's a project or there are students to be trained. I do this just because it's so much fun. So I'm just going to talk to you today about what is so special about counting butterflies. And later during Q&A, I might share some more information about how counts could be done and what we could gain from that. I'll also give you a few examples of the kinds of things that we have discovered just by counting butterflies all over India and also over the seasons. So I'll talk about that. Great. So this is the title of the talk I'm going to give today, Counting Butterflies for Ecological Research and Biodiversity Conservation. Fortunately, the way we count butterflies is the same, no matter whether you're doing it for research, for pleasure, or to help conservation movement. So we'll launch into that. Fortunately, India is blessed with tremendous biodiversity. It's all around us. If you have not heard this before, you take up any city or town in India, we have many more butterflies locally than you would find in entire countries in Europe, for example, where a lot of work has been done. So it's great that we have so much biodiversity. On this map, I've shown these 4 biodiversity hotspots that occur in parts of India, and these are spaces with fantastic biodiversity and especially butterfly diversity. But you don't really have to go very far to see butterflies. You can actually see butterflies in your backyards, in your nearby urban park. You can practically see them anywhere. Our agricultural fields have many more species than you will find in many other parts of the world, especially the temperate regions. So it's fantastic that we have it. So then the question is, why do we care about all these butterflies and nature in general? The first thing I will say once again is that we should do this because it's one of the most enjoyable things that you will do. Not necessarily because there's something to be gained outside of that, or there's some purpose beyond that, whether it's helping science, whether it's helping conservation. I think you should do this just because it's so much fun, as I mentioned earlier. But going beyond that, if you want to ask slightly larger questions, or if you want to help something beyond yourself, then I'll point out that components of biodiversity, including butterflies and especially their caterpillars, are crucial for ecosystems and habitats around us to function properly. They're also important for pollination and crop productivity, for example, in many areas, and for certain kind of crops, of course. Most butterflies indeed are not damaging to crops. They might be mild pests sometimes, but most of the butterflies are actually completely harmless to most of the plants that we grow around our cities, towns, and agricultural fields. Monitoring biodiversity can tell us how healthy the environment is, how healthy the ecosystems are, whether there are too many, whether there's too much disturbance in the habitat, and so on and so forth. So butterflies can act as indicator species which can tell us about the environment like only a few other groups can, because they're easy to identify, they're easy to observe, and therefore they're easy to monitor, which may not be the case for very small insects or very small plants, for example, which may also be very hard to identify. The next point is that carefully planned and implemented long-term monitoring can really help inform our national as well as regional policies. And wildlife managers, for example, state forest departments, can benefit tremendously from the information that we gather. And you don't have to be a scientist to do that. In fact, in Bangalore and in Pune and Delhi and several other cities now around the country, forest department actually depends on a lot of naturalists and local NGOs, nature-related NGOs, to provide them information, to provide them assistance in conserving local habitats as well as local species. So this type of information not only is useful for scientific studies, it can actually have real impacts on how ecosystems and habitats are managed, as well as how they might be enriched for our local native diversity. And of course, long-term data sets make biology very interesting. And this is something which is a little bit strange to me when I hear from people, that people often ask me, why do you study this? You know, what is the use of butterflies for us? It doesn't necessarily have to be something that is useful to us. It could also be something that makes it very interesting for us to look at nature. And the very fact that so many of you will be looking at this video, at this presentation, is a testament to that. Second thing is, whenever you publish any scientific paper, whether it has any tangible implication for the well-being of people, or agriculture, or whatever else, people contact me. Often our research papers and their findings are featured in news stories, which tells me that people are genuinely interested in knowing about nature. If they have been exposed, if their minds have been broadened, they definitely want to know how nature ticks. And butterflies being so colorful, butterflies being so charming, they are particularly a big attractant to a lot of people's curiosity as well as their interest in nature. And the second last point I want to make is that monitoring programs can uncover biological patterns, the long-term or very large patterns that are very hard to see at local scales. But when you zoom out a little bit, and when you look at really large data sets, then this becomes really obvious. And one of the most important example of the conservation, social, and scientific breakthroughs that such monitoring and counting of wildlife has done is the understanding of climate change. If you look at the history of our understanding of climate change as well as its importance for conservation, you will see that there were two groups of organisms that were being monitored on a regular basis, not because scientists or naturalists were interested in trying to understand what is the impact of climate on these organisms, but just because they wanted to see how their numbers keep going up and down. And through the years, and sometimes after decades of work, people slowly started realizing that the time when birds nest, for example, or butterflies start appearing after the cold or after a rainy season, has started shifting. And then slowly it was realized that this is linked to, for example, amount of carbon in the atmosphere, or other climatic fluctuations that happen, or other climatic trends that we now know pretty well. But all of this was uncovered because there were people, naturalists, as well as professional biologists, who were going out and counting birds and butterflies for not only years, but decades. So it's really, really important. And it's only a matter of time before such large data sets and such large initiatives to count organisms really starts showing something that we otherwise wouldn't have discovered. And it's especially important for India because we are still a country which is an informational black hole for most of the things to do with nature, and especially to do with natural history or biology of organisms. We still do not know basic things about a lot of our species. So we really need to start counting, start observing, so that we know our nature, our biodiversity really well, which will only help us in the long term, not only to appreciate nature, but also help it survive all the developments that are going on, especially in a country like India. And that is something that we all would want to see. So this is also not something that is starting now. People have realized the importance of doing this for a long time. And one of the most celebrated schemes, a monitoring program in this area, is the UK Butterfly Monitoring Scheme, or called UKBMS in short. And they have been monitoring abundance of UK butterflies since 1976 for more than 40 years now. And over this entire period, they have more than a quarter weekly visits to more than 1,500 sites. You can see the map of localities of the UK there, where people have been going and counting butterflies for a long time. When you consider the entire effort that has gone into these 40 years of counting, people have walked more than 500,000 kilometers trying to count butterflies. This is, of course, over the same paths and stretches of paths that have been identified for the purpose. But when you count up all the walking that has been done, it's more than half a million kilometers, which is just astounding. Generally, the UK BMS numbers are just staggering at this point. And the total number of butterflies that have been counted is nearly 17 million now. So that's 17 million individual butterflies, not species. Species are much fewer, less than 60 in fact. But because it's a long-term data set, the number of individuals that have been counted is enormous. And this has shown some really interesting patterns. For example, you see here at the bottom now that if you consider from 1970s to 1920 now, or rather 2019, which is when the analyzed data set ends here, you see that over 10 years or longer periods, the butterfly numbers have changed quite a bit. So on the x-axis here, the numbers that you see 0 to 50 show the change in numbers. They're not the exact number of individuals, it's the change. And of course, on the y-axis, you see the years. So, and what is striking is that the patterns that you expect, they're not only seasonal, meaning in different seasons of the same year, you might see changes, but over multiple years and multiple decades, you will see fundamental shifts in the abundance of butterflies, the number of species as well as the number of individuals that you will see. And this really you could not have dreamed of. You could not ever have suspected that butterflies might show such long-term population fluctuations. And what is interesting is that about 30% of the butterflies, UK butterflies, have shown declines over this 40-year period, and only about 23% has shown an increase. So often the total number of butterflies that have been counted may not differ much, but critically important species, more sensitive species, typically they are the ones who have been declining, and species which can survive in more human-dominated landscapes have been doing a little bit better. So all these things you really couldn't have figured out from any other kind of work. And this started from 1976, but this has really spread throughout Europe now. There are national butterfly monitoring programs in more than 10 European countries, and they're providing unparalleled insights into population dynamics over continental scales. And in the US also, there is this grid-based system which has been going on for some time, and the 4th of July butterfly counts. So the point in showing you all of this is that UK, for such a tiny country, so few people compared to, let's say, what we have in India, just by persistently doing something and doing something well, they have achieved something of a legend. Just by, I mean, you look at the numbers, they're just unparalleled. You just do not see that kind of work almost anywhere else. And they've done it for birds, they've done it for a number of organisms. I'm just showing you numbers for butterflies. So there's a lot to learn and there's a lot to do in our country. There's other kinds of monitoring. For example, this person shown here, Professor Daniel Janzel, he's a professor in University of Pennsylvania. In the US, he has been raising caterpillars on plants for the last 40 years, or slightly more than 40 years, perhaps by now. And he has discovered an amazing number of things just by doing this one thing. He has been doing this year after year, decade after decade. So you don't necessarily have to count only adult butterflies. You could be doing any number of things. And anything you do ultimately is going to tell you something very interesting about whatever that you're counting. In India, unfortunately, the history is both short and also fairly shallow. The first known monitoring that was done, as far as I know, was something that I started way back in 1992. I was 19, by the way, at that time, which is probably younger than most of the viewers today, which goes to show that you really don't have to be a professional or an expert to do this. You just need to start. And if there's anything that I want to achieve today, is for you to start. Once you start, you will learn a lot of things, no doubt about it. Once you feel like you want to do this, once you understand, once you realize that you really enjoy this, then of course you will learn all the things. But I'm going to tell you something about the methods as well and some of the cool things that we have discovered. but really, I want you to get started. And from my example, I can tell you that it's not hard to start. And this was at a time when making a career in ecology, doing research was not really something that was discussed a lot in the society or the family that I grew up in. So you are at an advantage. You already are passionate about nature. You probably already know butterflies well enough, at least in your area, that you can start very easily. And I also managed to publish a paper based on this. That was my first paper. And that's the first paper, as far as I know, from India, based on this kind of seasonal monitoring of butterfly populations. And this work showed that fire and grazing affect species composition. In other words, which species occur in which areas and how, in what numbers. That is affected by these two anthropogenic disturbances. And predominance of herbs versus tree cover, which is again affected by fire and grazing, also affect flight periods, meaning when butterflies are flying around as adults versus, let's say, caterpillars in a dormant stage. That is also affected quite a bit. And since then, once I became a professional, first as a PhD student and now as a faculty member, we have been counting butterflies using the 30-minute count method that I'll talk about later. And this is a large network. I've said Kunte and colleagues, it's more colleagues than Kunte, but I've put myself there just because we have developed this 30-minute count method, which has become quite popular now in certain groups. And one of the groups that has had the biggest impact and the longest record of doing this is the group that has been doing butterfly monitoring in Bengaluru. This started in 2012 when I came back after my postdoc, and I happened to meet Rohit Girotra in Bangalore, who had found a butterfly he could identify. So we met up to look at that butterfly. And I mentioned to him, why doesn't he start counting butterflies? And that is how this started in early 2012. And now this has been running continuously for 9 years now. We are in our ninth year. And this group has counted more than 90,000 individual butterflies just from this one place, Doresanipalya Forest Research Campus. But other students and people have also been counting butterflies in places like Bangalore University campus or GKVK campus. And I just mentioned the Doresanipalya effort, and we have been using 30-minute counts for this since the beginning. And now people have started doing this, I think, for the last 2 or 3 years in Mysuru and Pune as well. And this is the longest running effort. The data are being analyzed. Some of the interesting things that we have already found out, for example, is that if you look at the data from 2013, 2012 to 2019, from 2013 to 16, there was a fairly sharp decline. On the y-axis, you have number of individuals seen per year. So in the entire year, we counted how many individuals were seen. And from about 16,000 in 2013-14, we counted just less than 10,000 individuals by 2016-17, with the numbers going down fairly steadily. And that started worrying several of us. And reporters started calling me saying that they had heard that numbers were going down. Was it because of climate change? Was it because of urbanization? And I kept saying, don't jump to conclusions yet. These things may be true, but we don't know yet. We just have to look a little bit longer and then see what happens. And sure enough, in 2017-18, the rains were really good and the numbers went up. 2018-19, again, rains were not that great, but 19-20, rains were fantastic. And this year, again, rains have been good. So we expect that the numbers are going to be fairly high. So even if the habitat is surrounded much more now by urbanization, all the urban growth, the habitat itself inside that forest hasn't changed much. And what this graph shows is that butterflies tend to be much more tied with the climatic factors rather than disturbance around if the habitat itself stays more or less the same. And this might actually improve because the Karnataka Forest Department is in fact replacing some of the exotic trees that they had planted earlier. and planting more native biodiversity friendly plants. So you might actually see some improvements in these trends over time, and we'll be able to track all these changes as we continue to count. By the way, these data are collected every 15 days, every 2 weeks, which avoids counting the same butterflies every week. And 2 weeks is long enough that most butterflies would have laid eggs and would have died between any 2 counts. And in the second graph, you see that average number of individuals per 30-minute count has stayed more or less the same, with some ups and downs, again, probably because of the climatic variations. But the number of species seen per year hasn't changed much. We have consistently been seeing about 120 to 140 species per year. So these are all interesting patterns, and these patterns will become even more interesting as we have long-term data, not only from Bengaluru, but also from other parts of India. And then there have been a bunch of other studies. I won't go through each and every one of the points I've listed, but suffice to say that a number of master's students and PhD students in the last 2 or perhaps even 3 decades have collected a bunch of data on butterfly population dynamics through seasons, mostly not across localities so much, but through seasons. And some of these have really been nice studies which have revealed something interesting about the biology of the butterflies, and we'll talk more about the 30-minute count soon. Another thing that has been looked at is population dynamics of this small butterfly called plains cupid. And here you see it in the middle, you see the wet season form of the species, and on the 2 flanks you see the dry season form. They look quite different, and here is the seasonal climatic changes in temperature, relative humidity, and precipitation. And there's no surprise, we all know that these 3 climatic factors do not stay the same over the years, over the seasons. But what is interesting is that we saw on the y-axis here, it says number of plains cupid numbers, and here you have proportion of dry season forms in the number of individuals that were seen. And this is of course January to December, and what you see is that the numbers are quite good in during the monsoon and right after the monsoon. During the summers, the numbers are quite low, which is a pattern that you will see in majority of the species, although some species are much more common during the summer. And this is again from what we have counted in the last about 20 years. But the number of dry season forms is much higher. Almost all individuals that you see are dry season forms during the summer, and most of them during the winter. So these are interesting patterns, and now you understand why is it that these forms change. And we were able to do some very nice study looking at the climatic factors and how they affect butterfly numbers. For example, in this case, you see that if humidity is less than about 43%, then there are very few butterflies as counted per week. If the relative humidity is more than 43%, then you see a lot more butterflies. For example, here about 15 individuals per week, and here more than 9 individuals per week. And after humidity, the second most important factor is temperature. If the temperature is greater than about 28 degrees Celsius, then you see fewer butterflies. And if the temperature is less than 24 degrees Celsius, that's average in a day and in a month, then you see that the number of butterflies you see is a little bit higher. So if the humidity is greater than 43% and if the temperature is less than 28%, then you see the maximum number of individuals of this butterfly. So apart from just knowing when these butterfly numbers go up and down, now we know, especially in case of this particular species, and this was near Nagpur in central India, now we know what are the specific climatic factors which affect how common a butterfly is going to be. And again, these are sorts of things that you will know only by counting long term, whether it's through the seasons or over the years. Here is another interesting study of the cabbage white butterfly done by a master's student in NCBS, which shows that these butterflies And I don't know, this is a very common butterfly. You go to the Himalayas or to Uti area, you are going to see this. It's very, very common. And I don't know how many of you have noticed that their undersides are quite variable. Sometimes you might find individuals with very pale undersides like this one, or very dark and coloration with a lot of melanin deposited on the wing in other individuals. And Shubham Gautam, who did this study, he was able to show that if you look at the amount of wing melanin in the wings, especially the underside, hind wing underside, there are two patterns. In winter, butterflies are much more dark. You can imagine it's cold, and therefore they need to heat up a little bit better. And of course, they heat up better if they're darker. A fairly common phenomenon in animal world, which you have heard of. But now it shows that winter individuals tend to be much darker compared to summer individuals. And then within that, males, which are these solid circles, versus females, the plus symbols, you will see that there are much more pluses on the higher side and much more rounds on the lower side, meaning that females generally tend to be darker compared to males. And there are several studies showing that darker females are able to remain active for longer and then lay more eggs as a result of that. So this study nicely shows that if you monitor butterflies through summer and winter, you will see much darker individuals in winter and also much darker individuals at higher elevation. On the y-axis, you have elevation in meters, and you will see that the trend is upward, meaning that as you go higher up in the Himalaya, you will see that the butterflies are darker. And again, this sort of really interesting insights into the biology and morphology of butterflies can only be gained through this sort of studies. Okay, so how do we actually measure, how do we count butterflies to find out all these interesting patterns? Oh, by the way, in case of cabbage white, this study was also important because the Himalaya, as we know, are changing because of changes in climate. The entire ecosystems are changing. You might have heard that many of the glaciers have already melted, and that is going to have a huge impact on water availability and a whole bunch of other things. So we want to know how butterflies respond to all these changes, whether because of warming trends, they will change in their morphology or in their behavior, or both. And the cabbage white study was quite instructive for that purpose. So how do we find out all these interesting things? And I guarantee you, there are going to be interesting stories happening right in your backyard. You don't have to go to remote places. You don't have to do something special. If you just keep counting in your backyard, as many of these British citizens have been for the last 40 years, you are going to find something really exciting. So let's look at how organisms are counted, and the methods that we are going to look at are fairly widely applicable to a bunch of organisms, but of course we'll consider butterfly examples a little bit more. And I just realized we are about 30 minutes into the talk, so I'll try to be concise a little bit more now that I hope I've got you hooked onto counting butterflies and understanding the importance of why it is crucial to do this. So there are three main types of sampling methods. One is line or belt transects. These have a defined space, a certain width from the transect line. So the blue is the line on which you will walk. Typically, it's a straight line. And then you count butterflies, for example, let's say for 5 meters on the left or right of you and 5 meters above you. And of course, you have a certain length defined, let's say 500 meters, and you count every butterfly in that area. The second method that is used quite commonly is point counts. And this might be true under certain circumstances, which I'll mention later. But basically, you stay in one place and you just look around and see all the butterflies that you see around you in a given time, let's say half an hour. And some butterflies may be seen a little bit closer, some butterflies may be seen a little bit further. You note down the distance from you, but then you define the radius that you will consider around you in which you will count butterflies. And then the third method about which we're going to talk quite a bit, because it's quite a nice method for our certain purposes. So we'll talk much more about that method than other two methods that I've just mentioned. And that is called time constraint counts. We're going to call it 30-minute counts because the time is constrained to 30 minutes in our method. And I'll tell you the reasons for that in a bit. So basically what you do is you don't worry about what kind of path you take. You walk along whatever path that is convenient or whatever habitat that is convenient to you and feasible to you. And then you keep counting butterflies in 30-minute slots, and every green line here tells you how much distance you have covered. So distance doesn't have to be the same across every 30-minute count. But you do have to organize all your numbers in these 30-minute slots, which is why it's a time constrained method and not a specially explicit method, meaning that space is not defined, space is not needed to be constant, but time is needed to be constant across across all the counts. So in line of belt transect, what is important is that it's a rigorously developed and tested method. It has been used for a long time, for decades in fact, and it's been used for large mammals and forest trees, for example, quite a bit. The statistical methods required for this are highly refined, and these are suitable for a wide variety of scientific purposes. And therefore, this has been one of the most important methods to count all sorts of organisms. There are no long-term studies which have been used with this for butterflies, for obvious reasons that I'll mention later. But you can estimate density as well as relative abundance with this method, which, for example, you can't do with 30-minute counts. You can only measure abundance, but not density. The disadvantage of this is that it's temporally and spatially explicit. So temporal dimension here is regarding time, and spatial dimension is space. But I'll be using these terms again and again, so I thought I should define them right now. But because they're explicit, they're too rigid for a lot of studies, for a lot of purposes, and in a lot of landscapes. So that rigidity is a strength if you want to do highly rigorous scientific studies, but it's also a limitation if you want to be a little bit more flexible for the reasons that I'll mention on the next slide. And therefore, they're not this is not suitable in uneven and difficult terrain. For example, most Indian forests, especially in the hills where you cannot walk straight, and you cannot expect to cover the same amount of distance in every chunk, because it's just impossible. Sometimes it's going to be too steep, sometimes you have to wade through waters, and so on and so forth. Sometimes you're really going to see lots of butterflies in one place. So you're not going to be able to maintain constant pace, which line transects depend on. So for a lot of Indian conditions, this is not really a very good method. And if you really want to do it rigorously, you also need to plan well in advance. Ideally, you want to lay random line transects on a landscape from a map first, and then go and count butterflies there to be truly objective in choosing your spots. So it does require substantial advanced preparation, which has its advantages, but a lot of disadvantages as well. And as I mentioned, it's not feasible in many tropical areas, not just Indian landscapes. And it's also not very good for citizen science projects, because most people cannot handle the kind of rigidity that this method requires. And that's one of the reasons why people haven't really used it so much for birds or butterflies. And I keep mentioning birds because a lot more data exists for birds than it exists for butterflies, although slowly that might change. Butterfly observers might catch up on a lot of number advantages that bird data have. Anyway, so point counts, as I've mentioned, what you basically do, it's a fairly rigorously developed and tested method, has been used for decades, especially for areas where either organisms are unevenly distributed or where movement is hampered, especially if you're sitting in a tree canopy, you cannot walk around, right? If you are good at climbing trees, you can get up in the tree, but then once you're there, I mean, a few feet or a couple of meters on a very large tree is all you can manage. There you're basically bound to one spot. So there, point count may be the only method that you could use. And there are fairly good statistical methods available for this sort of data because this has been used for quite a lot. But these are typically good for only single species studies for butterflies. It's a bit tedious for butterflies and it's usually not productive for scientists or attractive to citizen scientists, and this hasn't caught up at all. But I wanted to mention this precisely because some kind of studies, even on butterflies, may be best done using point counts. But the method that I have come to favor over the last 25 years is the time constraint counts because it works for Indian conditions. And that is what I'm going to spend the next 5 to 10 minutes talking about. So I'm going to start calling it 30-minute counts now rather than time constraint counts because 30-minute is the unit we have chosen. And we have chosen that unit because if you look at most Indian habitats, 30 minutes is a reasonable amount of time to cover any particular habitat. Beyond that, the habitats may be too small and therefore you may not be able to cover them for more than 30 minutes. And maybe you have a single patch which you can cover in 30 minutes and therefore you don't want anything longer than that. But if the habitat is longer, let's say you're in a big urban park, I'm pretty sure if you go to any big city, there are going to be urban parks large enough where you can take 2 or 3 or even 4 counts each of 30 minutes. And if you go to, let's say, a wildlife sanctuary or national park or any wild place outside of cities and towns, there you can go on for the whole day and you could organize all your observations in these 30-minute counts. So before I assess the strengths and weaknesses of this or limitations of this particular method, I'll describe what it is because most people do not know, they're not familiar with this method. So first I'll describe this and then I will assess its strengths and limitations. And of course, we'll discuss if you have any questions right after I mention all the necessities of this particular method, and then I'll speak a little bit more about what you can do. Okay, so you should start counting when it's sunny, unless you want to sample butterflies which are active at dusk and dawn. Walk along a path, stream, wherever you will find butterflies or you know you will find butterflies. Note down the beginning time. For example, you could have a count from 8:30 to 9 in the morning, from 9 to 9:30, 9:30 to 10. So you could just line up your counts one after another and keep counting without a break. If you want to take a break, you can take a break for whatever interval you want and then start your next 30-minute count. At the beginning of every 30-minute count, note down the full date and specify date and month. Don't just write, as I've shown here, 10/9/20, because whether you're using British system or American system, 10/9 might mean 10th of November, 10th of September, or it might mean 9th of October, right? It becomes confusing, and it's going to be even more confusing if the year is less than 12, then you don't know what system you have used, and this becomes a big problem as time goes by, because if this is not recorded, then people don't know really what counting, what date system you have used. So you should either write 10 September 2020 or use full year, month, and then date, which is quite standard now in a lot of large data sets. And of course, as I mentioned, organize everything in 30 minutes slots. Mention also the general locality, for example, name of the urban park or wildlife sanctuary where you're at, exact location where you're taking that particular half-hour count, habitat type in which you're walking, relevant climatic conditions, whether it's sunny, clouded, windy, slightly breezy, maybe slight drizzle, and so on and so forth. And of course, number of people in the group. It makes a difference how many individual butterflies you will spot, depending on how many people you have in your group. There are more people in the group, there's more disturbance where you're walking, and therefore you might flush out more butterflies, and there are more pairs of eyes watching for butterflies, and therefore you're likely to see more individuals. So please note that. And then try to finish a 30-minute count in the same habitat. Don't cross over in a different habitat. So if you start in, let's say, an agricultural field before you get to a forest, beyond a certain parking lot where you might have parked, finish your half an hour in the agricultural field before you enter a forest stream, for example. And once you're in a forest stream, keep counting at least for half an hour before you, let's say, start walking on the slopes where the habitat is going to change. So stick to a single habitat while you're in that 30-minute slot, and then change over to another habitat if you wish. And this is again why half an hour is much better, because half an hour is long enough to have enough number of butterflies counted, but short enough that you are not stuck in a habitat, or you don't have to start mentioning from which point onward a different habitat starts within your count if it's longer than 30 minutes, which is a problem. So initially actually started with 3 hours and then 1 hour, and realized that was too long. And when we moved to 30-minute counts, since then things have been going well. So that seems like a very good uh, um, interval in which to count butterflies. The next thing is count all the butterflies that you see on either side or overhead. For now, count everything that you see at any distance. If you see a bird-wing butterfly flying at 100 meters, note that. If you see something flying, 20 meters above your head in the tree canopy, note it down. For butterflies which are very, very far, maybe you want to note down how far they were. But for other things, if you have time, note down distance. Otherwise, forget about it, because distance is not really a point of 30-minute counts. Time is the main unit by which you organize and you compare your data, and therefore distance is not necessary. And spend as much time in one place as you need to. For example, sorry. I think some text is missing. Maybe it's going to appear in the next slide. Look at the figure here. This is a landscape you're in, for example. What you could do is you start walking on a path. If there's a stream, you can follow a stream. You might want to cross over again with a path, and then continue along a stream again, and so on and so forth. So depending on whatever terrain you're in, you might choose your path carefully in the sense of whether it's feasible to walk on it or not. And notice that, actually I'm just going to do, I think the text is gone somehow, for example. So for example, what I was going to say is that these blue lines you see is a half-hour count in this particular figure. Yeah. And these orange dots are the number of butterflies you're seeing. As you will see, in this half an hour, you walk certain distance, you don't see too many butterflies. Then in this stretch, you see more, so the distance you have covered is less, because it's going to take you some time to identify a species, assess how many individuals there are, and then note it down. Similarly, here you will see there are lots and lots more butterflies. So there your half-hour interval in terms of distance covered is much smaller, but perhaps the number of butterflies that you count, the individuals you count, is more. So as long as you have the time measured properly, the rest of the things don't matter. I'll tell you what you could do to take care of space if you're keen on that. But otherwise, this illustration goes to show that time is really what you're counting butterflies against, not anything else. And I put this transect line here just to show that in this kind of landscape, it's impossible to put a straight line or a nearly straight line in any part of this landscape where you might be able to walk, let's say 500 meters in a fairly straight line without getting entangled into thorny bushes or really dense undergrowth or really steep slopes where you're just not going to be able to go through. And or if you're going to go through, there's too much vegetation and too much scree around that you're focusing on what you're running into rather than looking around to see if there are any butterflies around. So clearly this traditional transect method the belt transect method just does not work. Here, because the 30-minute count method is more flexible, you could still walk around where it's possible, where it's feasible to walk, and then count butterflies. So that's the advantage of this particular method. Now, if you want to bring in a little bit more rigor, and also if you care about space, which you may or may not, but if you do, then what you could do is carry a GPS unit, or on a smartphone install a GPS app and take down the entire track. And on this track, as you lay it down, you can mark locations of the beginning and end of every 30-minute count. So then you will know precisely how much distance you have walked, and then you could have some sense of not only how many butterflies you have seen in 30 minutes, but also in what span of distance that you have covered. So there are ways to include more rigorous estimates of space and other things if you wanted. But if you are doing this just for pleasure, if you're doing it in areas where space may not necessarily be important or feasible to estimate, then you could forget about that, of course. And of course, if you have the means, note down the temperature at the beginning and end of your 30-minute counts, if that's going to be useful in the long term. And certainly for data analysis, that's quite useful, because butterflies are active when it's fairly warm and humid. So if you have means to measure those two things, by all means do that. But don't worry about it if you don't have the means or the willingness to do it. If you're doing this just as a school project, for example, or if you're doing this with your kids as you take a stroll in your neighborhood, not necessary to worry about all these things that might be useful to a scientist, but not necessarily for anybody else. There, your 30-minute counts are going to be fine enough in the long term. Okay, so I will turn to now the advantages of this. Oh, before I do that, do you have any questions about how this method is implemented? Because this is what we are encouraging people to do when they start counting as part of the Big Butterfly Month. So if you have any questions about the method itself, let me know. Otherwise, some of the features that I mentioned here may be lost on you.
Dr. Kunte,
Yes.
there is a question in Facebook,
Okay.
which says, sir, my question, what about identification of species or genus level during the 30-minute count method?
So I think that was one of the texts that got somehow hidden. So if you cannot identify a species, you can mention the group. For example, you can say grass blue species. You can say brown crow species, and so on and so forth. If you can't even tell the genus, for example, in swifts, there are multiple species and genera of swifts. There are Caltoris, there are Panara, there are Pelopidas, and so on and so forth. And they're extremely difficult. Even I have to go, I or any expert, any level of experience cannot really help you. You have to go close. You have to sometimes look at the butterfly upper side, underside, and sometimes even specific brands to identify species. So in those cases, you can just say swift species. Typically, when we analyze data, we take out these records because they're meaningless if you're looking at species patterns. But if you're looking at slightly broader patterns, they may still be useful. So note down those data, don't ignore those groups. Note it down, just note it down as a group rather than as a species. Yeah. Any other questions?
Yes. Could you please let us know about how to omit the chances of repeatability in counting butterflies?
I do not know how I missed that one particular slide. I hope it's not coming up further somehow. But there's one slide on which I had put up both these things. Actually, a few more points. Now that you mentioned this, I remember the points I had put onto that slide. First is that if you're going to linger in an area for too long, for example, if you see mud puddling spot where you have counted butterflies, but then you want to spend some time taking pictures, for example. So taking pictures takes time. So if you are there for 30 minutes, I mean more than 30 minutes, you're probably going to see the same butterflies again and again. And because you can't identify individuals easily, you might note them down again and again. Every time you see it, every time it takes off and then lands again, you don't know whether it's the same butterfly or not. So you might want to note it down. So when you're going to stick around for a long time, also if you take a break to eat or drink, for example, at that time you should stop your 30-minute counts, take pictures, eat, whatever else you want to do, and then get back to 30-minute counts. So avoid staying in the same place where butterflies are just flying around and you might be confused about which individuals you're seeing again and again versus which individuals are new. Just stop there. You can still mention which are the new species that you're seeing, or in the 30-minute counts, if you had seen, let's say, only 3 individuals of a particular species. And then when you're having your snack, if you see 5 individuals of that, clearly there are at least 2 more individuals in that area. So you can make these notes at the end of your 30-minute counts, but do not continue counts. On the other hand, if you're walking on your regular count, you might be worried about counting the butterflies again and again, which is a problem with all the methods. That's not specific to this particular method. And there we just go ahead with the assumption that butterflies are not just going back and forth around you, and you're not just counting the same butterfly, same individual 20 times. So that's an assumption we have to live with. We can't necessarily know. And we can't even necessarily know that even for large mammals. For example, as Chital, if there's a group which is going back and forth, there's just no way you can estimate the precise number, even for such a large animal. Similarly, for butterflies, if you see a swarm, it's impossible to estimate exactly how many or count exactly how many individuals there are. So there, what you do is you just make a rough estimate of how many individuals there are. Let's say 50 plus, approximately 200. Considering that all the other butterflies that you will count will be only in ones and twos, or at best 4 or 5 at one spot, that versus 50 plus is already a big difference. So whether it's 50 or 60 or 100, you have to stop worrying at some point, because it's impossible unless you catch and keep every butterfly with you until you have caught every single one of them. You're never going to be sure about how many individuals there are. So there you just have to estimate. So there are these various things that you have to either assume or you have to estimate approximately, so that you have some data, not precise data. If you see one individual, there is no confusion how many individuals you're seeing. And if you can see them fly around, again, you can track them and there's no confusion. If there are several butterflies chasing each other and then sitting for some time, chasing each other other times, again, it's impossible to tell. Even if there are 4 individuals or 10, it's impossible to estimate if they're moving constantly and chasing each other. So do your best. You cannot always be precise. In ecology, especially when things are just going around, trees you don't have to worry about, they don't move, right? But for things like dragonflies, butterflies, you have to do your best, and your best is good enough in most cases. Yeah.
Doctor, I would like to ask one question.
Srirasha, could you please put the question in the chat box?
Yeah, sure.
Thank you. Dr. Kunte, the question for Atanu Bora, which says, do you think counting in different times of the day have an impact on the butterfly count?
Absolutely.
Habitats that are covered in the morning time have chances of having more butterflies than the habitats that are covered in the later part of the day. Is there any solution to make uniformity in counting?
No. There is no solution, because you're trying to put in everything into a format which might give you uniform data, perhaps, but that's you're not considering the biology in that case. So you should actually embrace this daily variation, daily meaning from early morning to late evening, because as Atanu very well knows, and probably most of our listeners today know, participants I should say, since it's more interactive, if you go out at, let's say, 5 o'clock, in certain parts of Northeast at 5 o'clock you already have sunlight, or 6 or 7 you're already seeing some butterflies. Yeah. But the species that you see at 6 o'clock, let's say, are very different from what you will see at 9 o'clock. which will be very different from what you will see at 2 o'clock perhaps. Between 9 and 2, perhaps the number of species that you will see will be smaller, but 2 o'clock might be a subset of 9 o'clock. But 6 o'clock and 9 o'clock almost guaranteed are going to be very different butterflies. So depending on what you want to do, you can either choose to count butterflies only during certain times of the day. For example, you can say in your particular area, 9 to 2 o'clock is when you see most of the activity. So you might want to just do your counts between 9 and 2, because rest of the time, you know, you want to be home, you want to do something else, you want to eat, and so on and so forth. But then you will spend the entire 9 to 2 interval counting butterflies. If you are in certain parts of the Northeast, Eagle Nest Wildlife Sanctuary, for example, right after sunrise, you have good sunlight and butterfly activity for, let's say, 2 hours. After that, it gets cloudy and very cold, and all butterflies, except perhaps Bhutan glories, go to rest somewhere in vegetation, right? Somewhere in dense foliage. So you're not going to see them. So the best time to count butterflies in Eagle Nest, for example, might be between, let's say, 6 to 7:30 or 8. And if you're lucky, if there's sun, you could count butterflies the rest of the day. So depending on where you live and whether you want to count every single butterfly, every single species in your area, you might want to decide when you want to count. For example, when I go to Namdafa, I'm like a starving person. I just starving for butterflies. I just cannot afford to take any break because there's so many butterflies everywhere. You go to Zero, Tale Wildlife Sanctuary, same thing. You go to many parts of the Western Ghats. There's so many butterflies the whole time. You do not want to take a break. Yeah, so I carry something small, or sometimes I don't even have breakfast or lunch. I will just quickly eat something before I go to the forest around dawn, and then I will come out only late in the afternoon or evening when butterfly activity has gone way down. And if I really want to photograph and see things like certain tree browns, for example, then I would actually be in the forest until I cannot see anything late in the evening. That is when certain butterflies come out. If you want to see activity of, let's say, Discophora sundaica, which is common duffer, you have to be out there when it's getting really, really dark. If you want to look at certain tree browns, for example, or Naga tree brown, you have to be out really late in the evening. So I would not say that you should change your 30-minute count method. You just count fewer butterflies, and in some intervals, you count 0 butterflies. So ideally, what you should do is, in your area, in every season, you should go on a few days, the whole time, before dawn to after it gets dark, and count butterflies in 30-minute slots throughout. Don't skip any 30-minute count. Yeah, and do that for at least 4 or 5 days, and that will be really good data. Then what you will know is, in your area, in that season, as you go from, let's say, 5:30 to, let's say, 6 o'clock in the evening, so 5:30 in the morning to 6 o'clock in the evening, in every 30-minute slots, how many individuals do you see and which species do you see? That will be a very interesting study as well, to see how species turn over through time of the day. And we have done this in Sikkim, for example. This was way back in 2008 to 2011 when I made several trips and we were out the whole day. We have done it in Namdafa, we have done it in a few other spots, but this needs to be done at a much bigger scale by everybody who wants to take count seriously. And everybody may not want to, but in any area, if you have a few people who are serious and who want to have this kind of data as well, which will be superb to tell people when to go out, right? If people are short on time, if you could tell them this is the time when you will see the most number of species, this is the time when you will see the most exciting species that you want to see. So go there, that will be really helpful, both for you as well as for any amateur naturalist who is just going there for pleasure, to photograph perhaps, but not to count, and things like that. And of course, for scientists, that's really valuable data, which shows how daily activity periods change with temperature, with amount of light, with whatever else that's happening in the day. So, and I know, Atanu, you are a very serious butterfly watcher. There are several others who are very serious butterfly watchers in the Northeast, in Western Ghats, some of you probably in other parts of India. So what I would urge is, please do this at least for 3-4 days in every single season that you can define in your area. You will learn something that nobody else knows about, and you will learn something that will in fact advance our understanding of butterfly activity, not only through the day but also over seasons a lot more. And of course, you will observe behaviors that you will otherwise not if you just go at certain times of the day and not others. Does that answer your question?
Yeah, that is answered. Okay, what Mr. What's it? Just a moment. Yeah, somebody has just written that the 30-minute count seems to be very complicated, and that's Sriharsha. He says, is it possible to repeat 30-minute count? It looks very difficult. I don't think it's difficult on the field. Maybe by looks of it, it could be difficult.
By looks of it, it looks difficult. If you want to simplify it, this is what you do. And this is what most people in Bengaluru, for example, do. Most of the Bengaluru Butterfly Club members who go to count butterflies every other week do this. So there's a regular trail that we have in Dorasanepalya, which is quite easy to walk, which is where you see quite a few butterflies. And then there are certain spots where you will see, for example, baronets. There are certain spots where you will see anomalous nawabs and so on and so forth. So once you know where you're going to walk, you just walk. If you go around with a camera or with binoculars or whatever else you carry on your nature trails, you just do that. You don't do anything special. The only thing that you do for 30-minute counts is note the time and note the number of individuals and number of species that you see in 30 minutes. That's all. Forget about everything else that I've mentioned. Forget about temperature, forget about time of the day, forget about absolutely everything. Go when it's convenient to you. Go when it's possible for you. You may not be able to go for 2-3 months to the field for various reasons. And then you may want to go to the field every single day for 1 week. Do whatever you normally do to observe butterflies. But when you're in the field, when you're writing down your notes, just note down the 30-minute slots, number of individuals, number of species in that. That's all. Yeah. And if you are in a particular habitat, of course, mention that you are in an evergreen forest versus, let's say, a teak plantation, or you are in a you're next to an agricultural field, for example, if you are going to other places, or you're in a wetland area, things like that. So simple notes that you write anyway whenever you go to the field. The only change you will do for 30-minute counts is do everything in 30-minute slots. Note down everything in 30-minute slots. That's all. You don't have to do anything else separately. Yeah. So it's not hard. You do it once or twice, you will think nothing of it. Yeah. This just comes naturally. A lot of us who have been doing this for quite a while, we keep doing this whether we are with other people or not, whether we are with kids or not, whether we are with other scientists or not. It just happens very naturally. So it's just a matter of doing this once or twice, perhaps with some experts if you are a little bit uncertain about this, if you are still a little bit afraid that you might do something wrong. I mean, you cannot do anything wrong. There's no concept of wrong when you're out there enjoying butterflies, right? So don't worry about it, just do it. And if you improve any of the methods, if you learn how to do it correctly after some time, you can again make a note that before this time, for the last 6 months, this is what I was doing. Now I've learned that this works out better for me, or in my habitat, in the areas where you go. And then make a note of that in your notes so that going forward, you know that there was some change between, let's say the first 6 months when you're counting butterflies versus after those 6 months. So don't worry about making mistakes. Don't worry about doing something wrong. It doesn't matter, because even if you do something incorrectly according to what other people are doing, you will still have enjoyed butterflies on your field trips. You will still have taken some notes, and those are still good for you at least. But from what I've seen, they're going to be good for even scientists. So absolutely don't worry about it. It's more in our mind. We are afraid that, we think that scientific study or this sort of systematic counting and data collection is something that people with experience do. It's not necessary. Anybody can do it. How we do it might differ a little bit. The quality of data that we collect might differ a little bit, and that's okay. Everybody goes through uh, different stages from being a beginner to being a very experienced naturalist. And many naturalists are better than many of the professional biologists. So eventually you'll get there. The important point, as I've mentioned right at the beginning, is to start. Just start, everything else will fall in place. It's easy.
The specific questions regarding 30-minute counts, I think, are over. There are a
Okay,
couple of general questions we'll take at the later stage,
sure.
maybe.
I have maybe 2 or 3 slides remaining. I'll go through those
Sure.
and then we can take more questions. It's already slightly over 1 hour. So if people want, I'm happy to stay on and answer questions. This whole evening I've earmarked for this, so I don't have any issues, but at least we should give people opportunity to leave if they are short on time. Okay, so very quickly, the method that I've mentioned now, it's reasonably rigorously developed if you include all the things that make it more useful for scientific studies. And if you do that, then it's fairly good, even for quality of scientific data that it generates. But if you don't want to include some of these things, as I mentioned, keep it simple, keep it easy for you, no problem. It's widely applicable in monitoring and studying butterflies or other things like dragonflies, for example. The methods are similar because they're very similar insects in terms of their activity and how we see them, how easily we see them. And this is especially important if it involves citizen science, citizen scientists, like the person who just asked a question. And the reason we prefer the 30-minute counts compared to any other method is precisely because depending on your level of experience, depending on your level of willingness to go a step further, you can do more or you can do the bare minimum, and it's okay. The 30-minute count can accommodate all of that as long as you have notes saying at what level you're working. It's fine. It's still good data. Yeah. So that's a major advantage because you don't need a lot of training, because you don't need a lot of experience in taking 30-minute counts. It's fairly good for citizen science projects. Whereas belt transects, you need a lot of training. You need a lot of experience in doing those things before your data become good enough to be used in scientific studies. 30-minute counts can estimate relative abundance of organisms. For example, you count the same patch, let's say in January, in April, in September, and then in November, you are going to see different number of butterflies as well as different species of butterflies. So because your 30-minute unit hasn't changed and the path you have walked has not changed, your data are still comparable. Even if you don't have any sense of space included in your 30-minute counts, because you are estimating the number in every single butterfly species in that area in that month, and the unit of your sampling, 30 minutes, is constant, you can look at relative abundance. That relative is important. You're not estimating absolute abundance or density. You're measuring and you're comparing relative abundance in one season versus another, in one area versus another, across one observer versus another, and so on and so forth. So that relative abundance is estimated really well by this method, and most scientific studies are fine with just relative abundance rather than density. It's very flexible. It can accommodate a number of real world scenarios, like a difficult terrain. You can avoid it, you can go around it, or you can take longer to cover that patch. But whenever you are in that patch, unlike in a belt transect, you're not trying to walk through that patch in a certain amount of time. You can take your time, look around, see what is flying around, note that down before you take any step further. So you can still accommodate a difficult terrain as long as you're fit enough to walk along that. Yeah. If there is a big swarm of congregating butterflies, for example, at a mud puddling spot, where you need to stop some time to notice all the butterflies that are around, and then identify them very well. If you go to Buxa or if you go to Namdafa, you're going to go crazy because there are going to be hundreds of butterflies in one spot. It's impossible that you're going to be able to identify each and every butterfly, not only in half an hour, perhaps in a morning. In Namdafa, for example, I've made plans several times to go, up to 19th mile or further, and I end up coming back right after, 500 meters, because you see so many butterflies in those 500 meters that you just cannot go past 500 meters in the entire morning. You can start at 7 o'clock. You come back around lunchtime, just 500 meters, doesn't take any time to walk back, but you have enormous amount of data from those 500 meters. So 30-minute count can accommodate that kind of highly clustered distribution, whereas transect, for example, it violates a basic and an important assumption of belt transect method or line transect method, and therefore that kind of density, that kind of congregation, that kind of concentration is highly problematic. Oh, by the way, there's one more point that I forgot to mention, that when you're counting, if you go along a path and you're going to return by the same path, you shouldn't be counting butterflies on the way back, because very likely you're going to be counting the same individuals of bush browns or rings or pansies and so on, which more or less stick to an area. So count butterflies only in one direction. So what you could do, for example, is make a loop. Go one way, come back the other way to the same spot where you're staying or where your car is parked, for example. And if you have to come back on the same path, count butterflies only one way, not the other way. The only thing you could do on the return path is note down additional species that you may see. So for example, if you have seen, let's say 30 species on the way towards your destination for that day, on the way back, if you see your 31st, 32nd, 33rd species, note down that species and the number of individuals that you see outside of the 30-minute counts. Yeah, but only note those additional individuals and species, not the same species, because you are very likely counting the same individuals of slightly sedentary species that don't move around too much. Whereas if you see, let's say, a red helen, or if you see what else, let's say white commodore, then it's quite scarce white commodore, then you're quite likely to actually see different individuals in the same patch, but it's best to avoid counting on the way back. Okay, all right. The last thing is, it's suitable in many tropical and mountainous areas. Any area in India is going to be perfectly fine for this. If you go to an urban garden, you can just walk along the path. If you are in an agricultural area, you can walk on the bunds created by farmers and not get into the fields, and that's perfectly fine. So it's quite adaptable in that sense, and that is a fantastic thing about this method. But then, of course, as I mentioned, it has a few limitations. You need to understand those limitations before you plan your studies, if you're going to be serious about counting. If you're not serious about counting to do your research paper or something else, then don't worry about the limitations. Just do what comes naturally to you, and that's going to be good enough. As I mentioned, you will have enjoyed counting butterflies. You will have enjoyed walking along these paths. You will have taken a lot of pictures that you will cherish, and that's good enough. But our experience in Bengaluru, for example, or even sometimes when we go to the field, is that people really get into counting butterflies. You know, they'll run back and forth. If the person who's noting down things in the notebook is a few hundred meters away, they will go back and forth to just say that some interesting butterfly has been seen, or the number of butterflies that they've seen. So people really get into this. So I don't doubt that most of you will get to a stage where you will want to know what you should do with the data, or to whom you should pass it on, so that it gets analyzed properly and we learn something from it. But do understand the limitations if you want to go any further than your own enjoyment. Another serious issue, if you are going to take the counts and give that into a larger database, for example, or if you're going to do scientific studies yourself, then you should know that statistical methods to analyze 30-minute count data are not as developed as, let's say, belt transect methods for statistical analysis, because they have been done for a much longer duration, and they have been used for certain kind of scientific studies for a long time, which is why the statistical methods are much more developed. But what I suspect is that because so many people are doing this kind of 30-minute counts, or any other kind of non-traditional belt transects, the kind of data that are generated, as I mentioned, 17 million records just from the UK on butterflies. And imagine if that starts to be done across Europe, across Africa, across Asia, the amount of data that we are going to generate is going to be so massive that it's going to be impossible for statisticians or scientists in general to ignore those data. And slowly these methods will develop. And if there are limitations, there will be several models that may be developed which will account for certain kind of uncertainties in the data, and so on and so forth. So these things will take time, but I have no doubt that these methods will be developed over time. But right now, those methods are limited. So you should know that. So you need to carefully think about why you're doing this and what you want to do with the data, if you want to do anything beyond just counting for pleasure or contributing to your regional repository, for example. But now I've changed the font in black, just to say that if you do want to bring in more scientific rigor, you can do that, for example, by taking GPS readings of the starting and ending points. You could restrict the length of, you could restrict the distance in which you measure, in which you count butterflies along the path, and so on and so forth, which will start to converge more on the belt transect method, if you want to be able to use some of the statistical methods used in traditional belt transect data. Or you could, for example, go to the same place again and again and count butterflies at the exact same time in the exact spots. So you could do a whole bunch of things. And if you're going to be a PhD student or a young professional trying to do this, then I will certainly recommend that you add these additional parameters in your data collection, so that you can use that in a bunch of statistical analysis, which you may otherwise not be able to do. For example, you won't be able to estimate density if you just do time but not space. But if you do, if you take readings on your GPS unit and define your distance from the line, then you could do some of those things. This is something I just mentioned, and this works. The great thing about this is that even if this is a relatively new method, this works. And this is from this study that we have done. This is my postdoc Sumana Tivli, Nitin Ravikantachari, and I. We have compared the two methods. Here what you see is green is the belt transect method, orange is the 30-minute count method, and purple is the 30-minute count method along a stream in an evergreen forest. Yeah. So you have that purple bar only in evergreen forest patch, not in scrub or urban, because streams of that kind were found only in evergreen forest. And of course, in evergreen forests, seeing butterflies was a problem because of a lot of shade. So if you look at, for example, number of species seen of habitat specialists, you see many more along the stream than along the path compared to, let's say, a belt transect, which goes through some shade and some openings and so on and so forth. So in some cases, actually, 30-minute counts give you more data. And other times, for example, if you're just looking at number of species or number of individuals in these three habitats, you consider all the species. For example, there are no statistically significant differences in the number of species that you encounter in 30-minute counts versus in a belt transect. So the two methods are fairly comparable in terms of the data you get, the number of individuals and the number of species that you count. But 30-minute counts tend to do a little bit better in getting more numbers because you avoid going through, let's say, very dense areas where you're not going to see butterflies and so on and so forth. So it's a fairly good method. If done properly, it is spot on. It can give you a lot of good data, both as a person who wants to do this in his or her neighborhood, as well as a scientist who might want to do it at a bigger scale. So that's that. That's basically the end of my presentation. I'll wrap up now by saying what you can do. It's a wonderful thing that we have the Big Butterfly Month. The count week is going to be from 14th through 20th. The Big Butterfly Month team will tell you more about it later, and once the Big Butterfly Month starts on the 5th. So there'll be more information eventually, but it's wonderful that you have this opportunity. You have a very large number of regional groups and national organizations have come together for the first time. So that's great. Probably the network is much bigger because of that. So go out, prepare your daily checklist in as many places in India as possible and count all the butterflies whenever you can throughout the day, if possible. And if possible, organize everything in 30-minute counts, as I mentioned. And that will be really, really helpful going forward. If we are going to do this long term, year after year, then it is important that we use a certain method which gives us data that can be compared. If you do a daily checklist, there's so many variables that are not recorded that comparing data becomes slightly more difficult, or the kind of conclusions that you can draw become limited. Whereas if you organize everything in 30-minute counts, you can compare it really across areas, across years, across people who are logging these data and so on. So please do 30-minute counts if you can. I highly recommend you to try it at least. If you do it a few times, you will see that it's easy, and I hope that you will continue. So please, please, please do that. And stay tuned in September for further announcements. Somebody has the microphone on. Can you please mute it? So in September, keep tuned in for announcements on Butterflies of India website or Facebook group for BBMI, as well as Butterflies of India, and any other social forums that you are active on. We are going to launch a butterfly app through which you will not only be able to identify species, but also take counts effectively. So look out for that, and there'll be further announcements about counting butterflies and monitoring butterfly populations as well. And these are all the NGOs which have participated in this, and I hope that there'll be more as the Big Butterfly Month starts. And thanks to all these groups for supporting this worthwhile effort, and of course Bengaluru Butterfly Club for work in Bengaluru, some of which I presented here, especially Rohit and some of my students who have been doing this in Doresanipalya in other parts of Bengaluru and elsewhere. So thanks to all these people for making this talk available and having refined this particular method quite a bit. If you want to learn more about the sorts of things that we do, or if you want to identify species, go to our Butterflies of India website, ifoundbutterflies.org. Any butterfly that you will see in your area is likely to be there. I'll be very surprised if you do not find your species there. We have majority of the species on the website, so you should be able to find it online. And we have amazing image gallery, so that will be a very good resource for you to identify your butterflies, to understand their polymorphism. And there'll be another webinar on how to contribute to this website in a few days. So I won't spend too much time on it, but it's a very nice website which will help you a lot in learning about butterflies and their wing patterns. And of course, if you want to read a little bit more about science, you can go to our lab website, biodiversitylab.org, and there you can read about some of the work that I presented today, and you can also read some of our papers in which all this information is given. So with that, I'll stop. Thanks a lot for joining us today. And I don't know what Shantanu and Sohail want to do, but if you want to give some time for people to put in questions or leave, maybe we can do that. Yeah.
Yeah, Dr. Kunte, that was an amazing and wholesome talk. I think you've covered almost everything that could be covered, but most importantly, the idea that it's easy should be understood. And once people go in the field and try and actually do it, I think it will be absolutely clear. It's one of the, like you said, it's one of the enjoyable things to do. Once you get a knack of it, you make a habit of it. It can really be helpful. So thank you.
I think that we should mention right now, Sohail, that all these groups that I listed earlier and that are listed on the poster, we are going to have organized events during the butterfly count week as well as other times. So if you're not sure exactly how to do it, just contact your nearest group, tell them that you're interested, go with them, and then you will see how easy it is to do this. And then you just need to start.
Exactly.
So Sohail will provide information about where to find these groups and where the contact information is, so that you can get plugged into these groups and then you can learn from people who are a little bit more experienced. We will be doing some counts ourselves in Bengaluru area and perhaps in other towns. So you can also write to us and you can join us if you're around the areas where we'll be doing these counts.
Yeah, so like you said, there are a few talks that are coming up, and 5th is our launch. And as we proceed towards the launch, we would be giving out more information about the count. The data sheet is going to be shared very, very soon. A calendar for all the talks is coming up, as well as we'll try and do more of these workshops so we can handle any more questions that are coming up in the future when people actually go and try out these accounts in the field. For now, we can do it in patients that have not been till now, and then have an open house if people do discuss any more of their experience and want to pick your brains. We can leave that at. So, Shantanu, do you want to come on?
Yes, I want to intervene here. There are a couple of questions that came in from Facebook because people out here are here only. Facebook questions, they mainly are like people are asking, do we have to maintain same time every day for count? Do we repeat the trails in different time slots? Or we do it, recommended to do it in the same area every time so the data we gather will not have impact of change or habitat change or terrain, types of food plants, etc. There's slight confusion out there.
Yeah, so I think I've answered all those questions before, but I'll just very briefly catch up on what I've said, on the advice that I've given. Basically, do what is convenient and what you really want to do, because ultimately, most of you are going to go out to watch butterflies for your enjoyment, not necessarily to count butterflies and not necessarily to contribute data to the larger initiative. So it's perfectly fine that if you want to count butterflies when you're out there anyway, at that time, sure, you can count. And therefore, you may not be able to go to the same place at the same time. You may not either have time or you want to bring in some variety or whatever else your reasons. You may not be able to do that, in which case, don't worry about it. But if you are a little bit more serious about this, for example, how the Bengaluru Butterfly Club and those people have been, then absolutely fix a certain time interval. We, for example, go out every 2 weeks, and we normally don't miss that at all. If Sunday is not possible, or on Sunday people want to go to another spot, then they might go to Dorasanipalli on Saturday, so that the 2 week interval is maintained more or less. So if you are serious about this, absolutely fix your places to go and space out counts in those areas every 2 weeks or weekly, whatever is convenient to you, or monthly or 2 monthly, again, depending on what is convenient to you. And once you do that, then other things will follow. For example, some of you might become more serious, and maybe you want to do this 9 to 1 every single time you go out, so that the time you are spending in the field in the same areas counting butterflies is again comparable, so that over the seasons and years you're comparing count data collected similarly in the same spots around the same times, and therefore those data become more comparable. So depending on what is feasible, what your interests are, how committed you become, I will leave that to you. And I think once you start counting, all these things become either clear, your commitment will become clear to you. Right now, you might think that you don't want to do this, or you want to do this forever, but your mind may change, your priorities may change, you might move to some other place. There could be a number of reasons why it may or may not work, and why you may change your mind to do this forever or to do it just once. So do it for a few times, and then we can have another session, perhaps later next month, and perhaps even in a few months once again, so that at that time you can ask me questions specifically based on the experience that you have before that, and then we can sort out your questions. And in some cases, I may not be able to tell you what you should do, because the solution may be very specific to your area, or very specific to your needs of why you are collecting data, or very specific to the kind of butterflies you see. So depending on that, we can at least discuss what I would imagine might work out. And then through discussion, you might come up with the best way forward for your area, for your counts. So don't really think that this is a scientific method which has to be done in a certain way. It's not a chemistry experiment where, a few ml of a certain chemical here and there, and there's going to be an explosion. Yeah, and people are going to die. It is not that kind of a method or a scientific endeavor where it has consequences if you do something incorrectly. So just be bold, just do it, and just do what you think works for you. And let's catch up in a few weeks, and perhaps in a few months once again. And who knows, if travel restrictions are lifted, I may be able to visit your area sometime, and then we can do a few counts together in the field. And at that time, I can also see how you do things. I can suggest a few changes, perhaps. We can perhaps come up with some improvements to this. So let's see how it goes. And then a lot of this confusion might be clarified, either because of your experience or because we might do things together in the future.
Thank you, Dr. Kunte, for that. Dr. Kunte kindly agreed to have a small kind of an open house with your questions. If you can kindly unmute yourself, ask your question, and then mute yourself pertaining to this topic of counts, you can go ahead now.
But after your question, please unmute yourself again. Don't forget.
You mute yourself, right?
Oh yeah, mute yourself after the question.
Do we have any questions?
I will also open my chat window so
Yes,
that if
please.
you have written anything, I can have a look. But I'll start looking now. So if you put up anything now, then I'll be able to read it. There are already lots of messages which I can't read yet. So I see the second last message is from Taslima Sheikh. She says, suggest separate Ladakh and Jammu Kashmir on IFB2. We'll do that eventually. I'm busy finishing something right now, but after that we'll do this. And I suppose everybody will have to do it now that the government has changed some of these boundaries. And
Somebody wanted an app on the 30-minute count some time back.
yes, that you will have that. Just wait for a couple more weeks, and we'll release that. And of course, we will put out the news everywhere so that you cannot miss it. And we are hoping to have this ready in time so that you might be able to use it during the count week as well. So we're trying hard to get that done. Unfortunately, we didn't have advance enough notification about BBM. I heard about it, I think, if I remember correctly, only about 2 weeks ago. So the app development was already in process. So it was supposed to have happened sometime now, but we'll just make sure that it absolutely happens, given that there's going to be a lot of it's going to be a lot of very useful for a lot of people. So we'll try to do that. All right. So somebody has said, please share the phylogenetic evolution of butterflies. That's a very different topic. So maybe we can have a webinar some other time about it. A very important topic, because phylogenies and systematic research has a lot to say about the names that we use. The way we treat our butterflies, whether species or subspecies or synonym. So there have been a bunch of phylogenetic studies that have happened in the last 15-20 years, which have fundamentally changed our understanding. But as I mentioned, that's a different topic right now. So let's come back to that in another webinar. If you have questions about counting butterflies itself, or if you want any specific advice about your area, about whatever else regarding counting during the big butterfly month, then let's discuss those things now. And once everybody is done with counting questions, then maybe we can turn to some of these other things that might be interesting to people. Next question. So Shantanu, you can also go through the
Yeah, I'm doing that.
See if there are any other questions.
If during a 30-minute count you stop to photograph or other distraction, do you stop the clock and add that to the 30 minutes?
So that's a little bit tricky. If you are in a group and only a few people are stopping to take pictures, or if you have to take turns, let's say there's a Nawab sitting somewhere and it's not going to move, so but only a few people can photograph it at a time, then you could take turns photographing it, but other people can continue walking and taking counts. If there's a situation where there's something absolutely showstopper that everybody wants to photograph, and then nobody's going to pay attention to any other butterflies, then surely stop your count. Make a note that you did your count for, let's say, 14 minutes, and then you saw something, so you have stopped the count, and then the remaining 16 minutes will start at a time that you note down. So you can certainly do that, no problem at all. As long as you make note of it, there's absolutely no issue with that.
Where can we record this data on count of butterflies? I use INAT for butterfly pictures, but this is much more data than what can be recorded there. Is there something similar to eBird for butterflies?
Absolutely, that is the app that we are developing. So you will absolutely have that, but it's going to be a little bit better than eBird. For birds, for example, you have to use Merlin for identification and looking at pictures, and then you use eBird app to contribute data. Our app will do both. So you will be able to use that as a field guide to butterflies, and you will be able to, of course, contribute data through that. So in the same app, you will be able to do both these things. So absolutely, that is precisely what we are developing, which is also why we are developing an app right now, because there are several other apps already available. Thiv Prakash, for example, has done an AI app, which is quite nice. It has limited number of species because you can only have so many images of species that AI can build upon. But as an app, it's done well. There are several other apps. So we needed not create another app, but we are doing this precisely because people will be able to contribute data much more easily. And even iNaturalist, for example, cannot handle that. So we will certainly make sure that our community becomes better by having programs regionally and locally, so that people who are experienced in taking counts will go and teach people in local areas and through
Okay.
regional NGO. And then of course we'll have the app, which can have a state list, which can have a regional list, let's say Western Ghats, so on and so forth. Um, so we can certainly accommodate all of that through the app. So just hold on, and of course, when the app is launched next month, we are going to release a smaller pool of species, because it's going to take some time to to um, develop all the species pages, all the resources in the app before we make it available. So don't worry about it right now. Eventually, we will have that. Yeah. So start using the app as soon as it comes out. Of course, unfortunately, if you're in Zero or Namdafa, then you're not going to be able to use the app right away, because all those species that you see commonly there will not be there at the first launch. But in a month or two, we will certainly have all the species that you will see in the most biodiverse places in India. So just download the app, and the next version will add those species. So you'll be able to use that for counting butterflies anywhere in India in just a matter of a short time. Yeah. There's another question here. It says, what about the written data? So written data, you will see that the form that is going to be circulated for Big Butterfly Month, it has an email address where you can email your data sheet. So just look up that sheet, and you will have some guidelines on collecting data as well as contributing data.
Okay.
Yes, we will be sending out the data sheet as soon as your talk is over. Once people have a grip on what to do, they can get to know the data sheet and maybe share what they understand and what they don't with us.
Yeah.
Right? Are there any
Yeah,
questions?
Pratisha has got a hand raised. Pratisha has a
Yeah,
question.
No, no, I don't know. No, no. Sorry.
Okay. There's a question of Dr. Udita Chakravarti. She asks, which counting method procedure should we follow for the plant-specific butterfly study in a forest ecosystem?
Very good question. So I mentioned Dan Johnson's study, and I told you that, for example, he has been finding caterpillars, and he has a big team on ground. He has trained a lot of Costa Ricans to do this work, so that he himself doesn't go and do all the work. So because he has this big team, they just go out, collect caterpillars, note down the plant on which caterpillar was collected, feed the caterpillar the same plant in their lab, and then when the butterfly comes out, they preserve it so that there's a sample which can be studied later. And then you have this butterfly and plant association, which has been noted. Every caterpillar gets photographed so that there is the larval phenotype, the coloration, the banding pattern, and everything is recorded along with the plant that it feeds on. If any parasitoids come out, they keep the parasitoids so that if anybody is interested in studying the parasitoids, they can they have the samples to study those as well. So depending on which kind of study you want to do, the method is going to change. If you're going to search for caterpillars, you could still do 30-minute counts. For example, you could walk in a patch and see how many plants, how many larvalose plants that you encounter. But a more meaningful method there could be, for example, you're randomly walking around in a forest or whatever habitat you're in, and when you encounter a larvalose plant that you're interested in, of the species that you're interested in, then you just look around that plant. If it's a small enough plant, you can look at each and every leaf. You can see how many eggs are deposited, how many caterpillars are there. And then you can take data down. For example, you can say, the plant height was so much, and the plant was in a certain kind of a microhabitat. Maybe it's in shade sometimes, maybe it's under the sun sometimes, maybe it's associated with rocky outcrops or something else. So note down all these things about the plant itself, and then note down, for example, how many leaves were on the plant, how many of the leaves were in the state that could be eaten by caterpillars. Certain caterpillars, for example, barren caterpillar, will eat even fairly old leaves. It doesn't mind that. If you feed old leaves of lemon to common Mormon caterpillar, it will die. It will not eat anything but fresh leaves, tender leaves, right? So depending on which species you're interested, the number of leaves that are available on the plant, the total number of leaves may be different from the number that are available to the caterpillar. So you can note those things down as well. So then your count unit is not time, it's not space, it is an individual plant. And then you will of course do this again and again in every month, for example, and you will note down how many, if there are leaf flushes to the plant, if there are certain times when you see caterpillars on that plant, and maybe for certain butterfly species, different species of plants are edible in different seasons, right? So they might breed on one plant in one season, they might breed on another plant species in another season. So as you start collecting data through time, through seasons as well as through years, you will start building a database of which butterfly feeds on which plant in which season and in which area. So if you have noticed, several people by now have published checklists of larval host plants used by butterflies in their area. I published my own list in first list in 2006, where I reported some of the same plants that were already known for the same butterfly species. But what was not known, for example, was that that species of butterfly feeds on that species of plant in that month and in that area. And that kind of information is really important. I'll tell you where it leads. There was a fantastic study, actually a series of papers that have come out of it. And that's again a 40-year data set. There's a very interesting set of studies that have come out of University of Texas at Austin. And I know this person really well. I was in his lab for some time, actually. His name is Professor Michael Singer. And for 40 years, more than 40 years, in fact, he has been going to the same patch of forest in California every year nearly. And every year he would go and catch some butterflies, put them in a small cage, or put them under an enclosed spot in the forest. And he will ask these female butterflies, which plant do you want to lay eggs on? So he'll give them a choice of plants in the same area and see how many eggs are deposited on which plant. And he did it year after year for 40 years. And what he has discovered is stunning, absolutely stunning. So over these 40 years, there was an exotic plant that was introduced. And the butterflies in this particular area, in the area of Yosemite National Park, actually, if you know where that is, and certain ranches around that area. So he found out that there was an invasive species, a non-native species, which was introduced in this area because of grazing. So grazing encouraged growth of this particular plant, and then butterflies preferred that plant because they could grow really well in that habitat on that plant species. And they completely lost the ability to survive on the native species, native plant species on which they fed earlier. So there was an evolutionary event where the butterfly population completely switched from eating one species of plant to eating an exotic plant. And then I think around 2007, 8, perhaps something like that, that ranch was bought over, and the National Park Services took it over, and they stopped grazing cattle in that area. And because the exotic was maintained in that area, because of grazing, that plant just vanished. And because those butterflies in that population had completely switched to the exotic species, they couldn't adapt back to the native species fast enough. So that population actually went extinct. And then over 3 or 4 years, I think, butterflies came from some other area, we don't know from where, but clearly from an area where they were using the native species still. And when those butterflies arrived in this patch, they could quickly find the native plant, which had already started growing quite a bit because there was no more grass and grassland being maintained for grazing. And therefore the native vegetation, the native conditions were returning, and the native host plant of this butterfly had certainly started becoming common. And this new population that came from some other subpopulation was already adapted to the native plant, and then they have started becoming common again. The butterflies started becoming common again. And all of this could not have been documented if this person had not been going to the same area again and again. So Michael Singer actually started this work, but Camille Parmesan and he have been doing this from 1980s together. And I think 2 years ago, if I remember correctly, they published a paper in Nature, which is one of the 2 top science journals. Absolutely groundbreaking research is published in these journals. And they were able to publish a paper in Nature based on this study because they had data over 40 years, through which they could show that slowly that butterfly had completely switched to the exotic species of plant. And the population crashed when that plant was certainly taken out, just in a matter of a few years because grazing was stopped. And then there was recolonization of that patch, and then re-establishment of that butterfly subpopulation. Absolutely fantastic study. So I'm glad that you asked this question, which was not about adult butterflies, but about the caterpillars and the plants that they depend on. And as this work also shows, you really have to choose your method for what you want to do. If caterpillar and caterpillar biology is your main reason why you're doing this, then you should absolutely go to the individual plant species. You can even mark those perhaps and keep going back to the same plant year after year, and then see how butterflies are utilizing that particular individual plant through seasons and through years. And of course, don't do this with just one individual plant. Do it with 10 plants, do it with 10 plants in different areas, so that you cover some geographical variation, you cover some soil variation, whatever other variation there might be. So if you want suggestions specifically based on what you want to do, we can have a separate meeting, but understand that 30-minute counts are good mostly for counting butterflies. If you're only looking at adult butterflies when you go on a walk in a forest. If you are looking for, let's say, parasitized caterpillars, you will have to use a completely different approach. If you're looking for eggs in certain times, you will have to use a completely different method. Hair streaks, for example, all those Chrysozephyrus and you know, that entire Zephyrus hair streaks group. All the green hair streaks are part of that. The water hair streak is part of that group, and so on and so forth. They're all found in the Himalaya, nowhere else in India. And of course, Himalaya and associated hill ranges in the northeast. So if you're looking for the caterpillars or eggs, which are found only in certain times of the year, they're not continually breeding like so many other species at lower elevations or at lower latitudes. So if you're looking for those eggs, you have to look for them only in certain months, only in certain weeks, perhaps. So depending on what you're counting, what the goal is, you will have to use a different method. The 30-minute count method is a more general method which suits most people, because most people just go looking for adult butterflies. But I really hope that more and more people will start looking at larvalose plants, at caterpillars, at parasitoids, at plant chemicals perhaps, which make it possible for caterpillars to feed on a wide range of plants in some cases and on just a single plant in other cases. Most people have shown or believe that this happens because of the secondary compounds that plants produce. And therefore, there is so much to understand there. As I mentioned, India is an informational black hole. And I did not mean just in terms of what occurs where. It's all this interesting biology of butterflies that we don't know, which is tremendously intriguing. If I start telling you stories, you're just going to be blown away by all sorts of things that butterflies do. For example, there is the coleus butterflies, the clouded yellows that we have in the Himalaya, and one species in the Western Ghats. That has certain parasitoids which are specifically evolved to parasitize only one species of butterfly. And the females of this parasitoid wasp are attracted to the mating pairs of the clouded yellow butterflies. So when these butterflies mate, the male leaves a mating plug inside a female. So this mating plug is basically a concoction of chemicals which the male passes on to the females, and that has a very species specific smell. So this wasp has evolved to zoom in onto that one particular smell. It will go and sit on the head of the female butterfly, and it will be carried around by the female butterfly after mating. And as the female butterfly is laying eggs on the plants where its caterpillars are going to feed, the wasp gets off from the head of the female butterfly and starts parasitizing eggs of the butterfly. I mean, how crazy is that, right? So all these things are absolutely amazing as biology. And of course, if you're a farmer, if you are an agricultural scientist, you want to know about these parasitoids so that you can control populations of butterflies or moths or whatever else is destroying crops. So it has everything from absolute curiosity that satisfies and people find quite amazing, all the way to applications that you cannot even imagine. So we should just go out and start observing butterflies and caterpillars and eggs and everything in their surrounding to understand them. And this is what I mean by informational black hole. Most of these studies are done on some species in Europe or North America or Australia, very few in India. With the number of species we have, with the amount of geographical variation, climatic variation that we have in India that butterflies face, you can bet that there are going to be amazing biological adaptations of our species to cold, to desert conditions, seasonal diapause, association with ants. We recently published a paper, my PhD student and I, absolutely mind-blowing work this student has done. And that is the first study of that kind anywhere in the world, not just in India. But we were able to find out something with our Indian species that everybody finds valuable. Anywhere people are interested in ant and caterpillar or ant and any other insect associations. So we have all these stories around us. We just need to look, and then we will have learned something truly remarkable, truly mind-blowing about our butterflies. And that is the information hole I would like us to fill. Not only which butterfly occurs where, but what does it do? What does it depend on? And this information is also going to be very useful. For example, you look at scarce jester, you look at black windmill, you look at any other species which is endangered, which is rare, which is endemic. You need this kind of information to help them survive, to help them persist in fragmented habitats. That is what we really need to do. So pick up projects. I can help you with methods, but today let's just discuss details of 30-minute count because our big butterfly month is coming up. But of course, contact me for anything else you want to do, and I can point you to the literature, point you to people who have done that kind of work, and then you can learn about the methods that they've used to do the specific study that they want to do. Yeah.
I guess we got time for one last question. Atanu Bora asks, what statistical method can be used to analyze point count data on butterfly migration?
Okay, so I will not talk about that right now because that's going too much into statistical details about which I don't know how many people are interested. But Atanu, please email me and we can perhaps set up another Zoom chat, uh, either just you and I, or if there's interest in a group, then we can have a more focused Zoom meeting for people who are serious about learning about statistical methods, details of some method, some new sampling method apart from 30-minute counts. And we can arrange that and I'll be very happy. I'm very, very committed to helping people start this sort of work, whether it's serious natural history or it's serious ecological work, serious conservation work. I want everybody who wants to do this anywhere in India to be able to do this. They should have the information, and your time, my time is your time. Just drop me an email and I'll be very happy to help you with whatever you need. If I'm not the right person to tell you about those things, I know everybody there is to know who's studying butterfly biology. I can put you in touch with the people who can answer your questions or who can help you in other ways. Just drop me an email. My email address is easy to find. If you search my name, my website comes up and you can just drop an email to that. It's krushnamegh@ifoundbutterflies.org.
So thank you, Dr. Kunte. I think that was an amazing talk. It covered everything there is to know about the 30-minute protocol. And to me personally, it seems really, really easy to do. And I'm just kicking myself that why I didn't start this count earlier. But now we have an opportunity. If there are all the questions are taken, we could end the meeting here. But before ending, I would just like to say that information on the count will be available on all our social media handles. You should subscribe to the YouTube channel and the Facebook page to get more information on the upcoming events and talks.
You will also hear about the app soon enough, I think.
Yes, and lots of exciting new things coming up. There's a launch also, and I've been telling Dr. Kunte that I've been waiting for this app for quite a long time, and very, very excited for that. As far as the count is concerned, we are going to share a data sheet for everybody we are in touch with as regional partners. And it is also going to be shared in all the other social media handles for people who are not, as of now, in touch with their regional teams. But we have almost all the states, 24 states at this moment. And if people who are listening today do want to connect and do want to participate, do write to bigbutterflymonth@gmail.com and we can direct you to your regional coordinators, and they can guide you in what is going on and how to take your contribution forward.
So if I remember correctly, we were also going to have a website, right? Is that going to happen soon enough?
Yeah, we're working on that.
Okay.
And yeah,
until then, we can also put up the form on I Found Butterflies or any other websites, so that
yes,
people have a range of websites from which they can download the form. We already have a page for Big Butterfly Month on Butterflies of India website. If you look at the top menu, there's Outreach tab, under which you will find Butterfly Month. What?
I think
Sorry, I can't hear you well.
Somebody's mic was switched off.
Okay. So we can put it up on I Found Butterflies website and any other websites, and you can just download it from the BBM page. And the I Found Butterfly BBM page is ready. I think iNaturalist page is also ready.
Yes, iNaturalist at India Biodiversity Portal also there is a project.
So we can just put it up on all the websites and people can download from there.
Absolutely. Yeah. So with that, I would like to thank everybody who was here, and of course, Dr. Kunte for giving us so much of his time and helping us out. We all want to contribute to butterflies, and we all want to contribute to conservation. and we're looking out ways to have more contributions apart from just lip service or Facebook or WhatsApp. We can actually start contributing data which is useful to scientists and do these counts regularly, which will help us understand so much more about what is going around with butterflies in our locality. So I hope this session was useful and we could reach out to most of the interested people. I would again suggest that if you have anybody else you think would like to participate and uh, if you would like to grow the family, please do get in touch with us and do share the information that you get along with everybody else. Is there anything, Dr. Kunte, you would like to add?
I will just want to say that, guys, within the next year, I really hope that I'll be able to travel all corners of India, and then we'll have an opportunity to count butterflies together in your area. And by that time, the app will be full-fledged, and I really hope that we'll be able to make a big move towards counting butterflies everywhere. Yeah. So I really look forward to that. And thanks again for participating today. And we'll hopefully have several webinars like this in the next few months. Bye.
Bye-bye. Thank you, Dr. Kunte. Thank you, everybody who was in here today. Thank you, people listening in on Facebook. Uh, have a good night, and we will see you very, very soon. Thank you. Thank you.